gastric cancer, but the doctors aren't sure yet.
Have they done any more tests?
The doctors mentioned something about biomarkers, but I'm not really sure what they are.
Well, why don't we go find out? I'm sure we could do a lot of research in the library.
All right. There you go. Hey, here it is.
It says here that biomarkers are used to indicate if there's an infection, disease, or if the human body has been exposed to something which could cause cancer, such as an environmental carcinogen. Most biomarkers today are still going through clinical trials, but many of them are successful in measuring accurate signs of specific infections and diseases.
Yeah, I believe he will be taking a few tests such as the carcinoembryonic antigen or the CEA test. It's a biomarker that successfully detects many types of cancer, including gastric cancer. Tumors have a high level of CEA, a glycoprotein, which is why this test can be used to successfully identify a tumor.
Yeah, and then if you keep reading, it says that CEA levels are still high after the removal of a tumor, and that can suggest that the cancer was not fully removed or that it has spread to other parts of the body. In general, if CEA levels are higher than 3 nanograms per milliliter, there's cause for concern.
Yeah, I think I need to do some more research online. Let me check it out real quick. Grace, look, here according to the National Comprehensive Cancer Network, biomarkers can be grouped into 3 different categories: gene testing, chromosome testing, and biochemical testing. The biomarkers used to detect gastric cancers generally use biochemical testing. Most biomarkers used to find gastric cancer focus on targeting microRNAs, epigenetic alterations, and genetic polymorphisms.
Hey, look at this video I just found. It describes the entire testing process.
It says a doctor will take a tissue or liquid biopsy to obtain a biological sample from the patient and test for any abnormalities produced in the body. They do this by staining the biopsied cells with one or several dyes that make the cells easier to see, and then they place the cells on a slide and view them under a microscope. A pathologist will examine the cells and will be able to see if the biomarkers reveal any cancerous or precancerous cells. If there are abnormal numbers of the biomarkers, the pathologist will see that the patient is showing evidence of cancer.
Wow, whoever made that video must have been really smart. This website here says that for patients who are concerned about cancer, doctors will typically use biomarkers within the biochemical testing category. This is mostly due to the multiple mutations that can be detected compared to the other 2 types of biomarkers, especially since gastric cancer is the 4th most common cancer in the world. A high success rate where the biomarkers indicate early gastric cancer could save many lives.
And then the National Comprehensive Cancer Institute says that most testing for gastric cancers involve non-invasive biomarkers that are present in blood or gastric juice. These tests evaluate the levels of antigens in the body, such as an increase in carcinoembryonic carbohydrate 72-4, alpha-fetoprotein, and CA19-9 antigens. Compared to a healthy patient, a person who has gastric cancer will have elevated levels of these antigens depending on which test is given and how far the stage of cancer is in the patient.
Oh, that makes sense. The book here says that the most common biomarkers used to test for gastric cancer are the combination of CEA and CA19-9 markers that monitor carcinoma of the stomach. But due to how sensitive and how close these tests are to other particular cancers, doctors will test microRNA, which involves 6 microRNA clusters and 21 microRNAs that look into the biological functions that affect the cell cycle progression and metastasis. These microRNAs can sometimes function as oncogenes or tumor suppressors, and they can impede regulatory processes in the body, such as growth suppressors and apoptosis. Because of this, dysregulated microRNAs are an indicator of cancer. However, even with all these types of biomarkers, they still pose some challenges.
Whoa, Ryan, do you even understand what you just said? I sure don't. I barely even know what an antigen is.
You're right, maybe we should ask someone. Who do you think would be a good person to talk to? Professor Scheib, just the person we need. Could you help educate us on biomarkers?
I'd love to.
So to clear up some of your confusion, antigens are foreign particles, usually proteins, that cause the immune system to respond when there is a harmful substance, which is in your body.
Carcinoembryonic.
Carcinoembryonic antigens are tests used in the blood that can help diagnose different types of cancers in the large intestines and rectums, and can also indicate gastric cancer. Elevated CAA levels in the blood are indicative of diseases, since it should only be produced in the gastrointestinal tract during the embryonic stages of development. Once a treatment has been implanted, they will continue to use CA levels to see if there is a treatment is working to stop cancer progression in the patient, such as carbohydrate antigen 72-4 is used to help predict if gastric cancer in the patient will be recurring and if it's better differentiated between benign and malignant tumors.
Oh, I see. Are there any other tests that use different antigens?
Yes, actually. Here, let me show you. So we have our alpha fetoprotein, or AFP. It is a blood test that's used as a tumor marker. AFP in adults shouldn't be at high levels. It is the main protein for a fetus during the first 3 months of development and then decreases. High levels APF in adults is only possible if there's some type of cell tumor or chronic active hepatitis in the patient. CA19-9 is another biomarker, and there's a high amount of CA19-9 found in the body and may result in the cancer in the body. CA19-9 is also similar to the carbohydrate antigen 72-4 and is also used frequently afterwards to track if a treatment for cancer is working or if cancer is worsening in a patient.
Wow, that background information was necessary because you sure had some big words going on over there. So I guess one of the main challenges is that many of these biomarkers that test for gastric cancer are still going through clinical trials because they can't always diagnose gastric cancer at an early enough stage for it to be treatable. Most of the markers are better used for determining what stage the cancer is in or how well a treatment is working, right?
Yeah.
The goal is to find combinations of these markers that are successful in the diagnosis of an early stage of gastric cancer, or there's still a chance to treat the disease before it causes major damage to the patient.
Well, Professor Shad, I certainly learned a lot about biomarkers today. Being able to detect cancer at an early enough stage is certainly valuable, especially when Americans have 1 in 3 chance of developing some form of cancer in their lifetimes. I know if I ever need to get tested for gastric cancer, understanding the whole process makes it a lot less frightening, and now I can take all this information to my uncle. Unfortunately, the survival rate of gastric cancer is around 31%. With the help of biomarkers, hopefully this percentage will continue to rise every year.
Ryan, you are absolutely right. And with modern medicine, doctors are making huge strides, not only in the study of biomarkers, but in the study of cancer as well. So in a few years, they might be able to detect cancer even earlier than they do now. A biomarker certainly has the potential to impact the medical world.
Do you think that biomarkers can tell me what grade I'm going to get on my biology final?
Yeah, how's that A looking with all the research we've done on biomarkers? I think the biomarkers indicate extra credit in our future.
Now, you two both know that's definitely not how biomarkers work. But since you guys asked, I say an A is probably out of the question.
That's fair.